All right, so let's talk about fermentation, which is a process that occurs when there's no oxygen present. Not all cells can do it. Not all cells can survive just doing fermentation. And we'll see why that is in a moment, but some can. So no oxygen, another word for that is anaerobic, meaning an meaning without, aerobe meaning oxygen. Anaerobic process. This is aerobic process. Note, there is also anaerobic respiration. So anaerobic respiration is different than fermentation. What this means is that it still uses ATP synthase to make the ATP. So it makes a lot of ATP from that. But it does not use oxygen at the end of the electron transport chain. So final electron acceptor, I talk about that in the oxidative phosphorylation lecture, but it does not use oxygen. So it would use something like sulfur. It would use something like maybe metal ions, maybe, things like that. So fermentation, basically what it is a process to keep glycolysis going. That's how I think of it. By regenerating NAD+. Why? So that glycolysis will still make some ATP for the cell. Some organisms only do this, fermentation. They don't ever do cellular respiration, aerobic cellular respiration. Yep, just fermentation. I say still because sometimes fermentation is like an alternate pathway to using the mitochondria if there's no oxygen. This is like very non-technical way to say this, but what it does is it dumps the electrons from NADH, dumps them into pyruvate or a pyruvate product, which is great because pyruvate is already made from glycolysis. So it's like to keep glycolysis going, let's just use what glycolysis makes already. So there's not additional steps in there. There's a conversion step, but generally it's pretty efficient. That end product is what you end up doing to make the same process go. So let's look at that. I'm just going to do this very generally. We've got glucose, and we have pyruvate, and this process up here is the glycolysis process, part of which is to make ATP, and that phosphate group, remember that's substrate level phosphorylation. So basically phosphate is attached to something, but there you go, gets picked up, okay? So this is like what we want to continue to happen. So how can we make that happen? The other part of glycolysis is this, because to make the ATP, you break the bond to kind of release some energy. When you break the bond, you can extract electrons. Classic or very key point. So you break the bond, glucose, you break it in half to make pyruvate, you can extract those electrons. So that's where you get your NADH from. Now, what do you need for glycolysis? You need glucose. So you either get that from eating, you get that from body stores, like body tissue, or which came from eating, or could come also from photosynthesis. So you got that as long as there's that coming in. You can see we need the ADP. Where does that come? The product after ATP use in the cell. So it's always being made because the cell's always using ATP, so it's always making ADP. That's not a problem. The deal is that it also needs NAD+. It comes from the electron transport chain after dropping off the electrons there. So when the electrons fall down it and they go from complex to complex, in the end they've got to get taken away, so they go into oxygen, and that ends up becoming water, and then water takes those away, removes the electrons. But what if you don't have oxygen? Then you can't transfer the electrons and it all gets backed up. Then what happens here is the NADH can't transfer electrons and you get stuck as NADH. So with no oxygen, NADH gets, think of it as stuck with its electrons. The electrons, its electrons. So if glycolysis is dependent on NAD+, and NADH gets stuck with its electrons, as in it's not making NAD+, then you have a problem with glycolysis. And so that's what fermentation does. It alleviates that problem. Fermentation is going to take NADH and bring it back into NAD+. And it does that using pyruvate. Like I mentioned, it's very efficient. Those electrons go and get picked up pyruvate, and the change in molecule there is going to give you lactate, which is the same thing as lactic acid, just in the acidic form where it drops the hydrogen ions. So that's your product here, your lactate, your lactic acid. This is cell types that do this. You've got muscle cells, which are, muscles are exclusively in animals. Certain bacteria do this. They produce lactic acid, which is like a sour taste. And so if the electrons get taken out, then you have NAD+, you can continue to do glycolysis and keep making ATP. This process here is that fermentation process. The other option, again, we want to regenerate that, and it's going to do a conversion step instead of just going into pyruvate. Other types of cells have different enzymes, so it does different processes. Why do they have different enzymes? Because they have different genes. So in something like yeast, pyruvate actually gets converted into a product, which I don't care that you know, but this is CO2 production. And then that product is where the electrons get transported or get transferred into that product. And that's going to make ethanol. And remember, as long as you keep on making the NAD+, you can continue doing this glycolysis, which is what's showing at the top, which is means that you can make ATP. What does this kind of process? Single-celled fungi called yeast. That's what yeast are. It's a big category. Okay. And the CO2 is what bread rises. The yeast are eating the sugar, doing glycolysis, and putting out CO2. And the ethanol, this is where the yeast in beer and wine, that's where you get the alcohol in there from the yeast doing this process as well. So if you want to do like beer brewing, you have to create a low oxygen environment. Why doesn't bread have lots of ethanol in it? A lot of it depends on how long you let the process go for how much ethanol is made and how much CO2 is made and such. Okay, so that is the basic idea of fermentation.